Hi everyone. So upon Isa's request, I'm going to go through the law of segregation and the law of independent assortment. They're very difficult to identify and more than anything to compare between each other because they relate to the same thing. So when a cell is going through meiosis, it's going to separate, or it's going to divide their chromosomes, their pieces of information. So in this case, this parent cell is super simplified because it's only telling you that this parent cell has two chromosomes. One of the chromosome contains an allele for a dominant trait, and the other one contains a recessive allele. Okay, so two chromosomes, and since they contain the allele for the same trait, let's say that this is hair color, they're going to be found in homologous chromosomes. That means that you have one chromosome that was given to you by mom and the other chromosome that was given to you by dad, and each one of them contain alleles. They can be the same alleles or they could be different. So dad's chromosome could have given you the allele for brunette hair for this trait, and mom's chromosomes could have given you the allele for blonde hair for this trait. It's the same trait, so it's homologous, but it's different alleles. So the law of segregation simply says that in homologous chromosomes, that means that chromosomes that contain the same genes, information for the same genes, the alleles are going to be segregated or separated randomly. So maybe this sex cell will get the dominant allele while this other sex cell gets the recessive allele. And maybe it could be vice versa. Maybe this sex cell would have gotten the recessive allele and this sex cell gets the dominant allele, but it's randomly separated. So these homologous chromosomes separate randomly, and the alleles for the same trait go to different sex cells. Okay, that's law of segregation. So again, if you can think about law of segregation as alleles, separation of alleles, then you are good. The law of independent assortment is similar to the law of segregation, but now we need to think about it as the independent assortment or the independent separation of genes, not alleles. So that means that you are now talking about non-homologous chromosomes because you're talking about different genes. So let's say that this homologous pair of chromosomes here have genes that code for big R, little r. Sorry, these are the alleles. Big R, little r would be the alleles. And let's say that this gene codes for eye color. So big R is brown eyes, little r is green eyes. Okay, now based on the law of segregation, this R, this big R, could segregate randomly to different sex cells, while this little r is going to segregate randomly to different sex cells. So each sex cell is going to get a different random, either dominant R, I said brown eyes, I think, and or little r, which are green eyes, I think I said. Okay, so that would be segregation. But in independent assortment, you don't only consider one gene, let's say eye color, you consider another gene or many other genes. Let's say in this other chromosome, there's the gene for height. Okay, dominant being tall, recessive being small. Okay, now these genes are not dependent on one another. Okay, that means that it's independent. It'll be randomly assorted. This big R will be randomly assorted to any of these sex cells independently of these big Y. Okay, so the genes are independent from one another, so they can be randomly assorted to sex cells in a very independent way. So if you want to see this here, for example, just by chance alone, this big R chromosome aligned in this side of the cell and this little R chromosome aligned in this side of the cell, and this big Y chromosome aligned in this side of the cell and this little Y chromosome aligned in this side of the cell. And because of their alignment, they're going to be divided through meiosis 1 and meiosis 2 into different sex cells. So because this big R aligned in this side of the cell, it becomes this daughter cell, this daughter sex cell, then in meiosis 2 divides into two sex cells, and each one of them is going to contain the big R allele. Since by chance alone this little r aligned in this side of the cell, same thing is going to happen in this daughter's sex cell that will then go through meiosis 2 and divide into the two sex cells with two little r's. Same thing with the little y aligned in this side of the cell. So same thing, little y in these sex cells. Same thing with the big Y aligned in this side of the cell. So big Y, big Y. But this is not the only alignment that could occur. If you look at this right over here, the R, the big R, could have aligned in this side, but then the little y aligned in this side as well. So now you have a new combination of alleles in your sex cells, in your gametes. If you see here, this one by chance alone got a big R and a big Y. But in this other arrangement, this one got a big R and a little y. So you can see that the trait that codes for the R's, big or little, and the trait that codes for the Y's, big or little, are independent from one another. They don't need to be arranged in the same way. They don't need to go together to the sex cells. They can go separately. They can be assorted independently. Okay, and this is very related to the idea of linked genes, because in this case there is no way that the gene that codes for eye color and the gene that codes for height are linked, because they are found in different chromosomes. Okay, but what happens when the gene of eye color and the gene for height is found in the same chromosome? They could potentially be linked, which means that in that way they are dependent on one another. They will likely, or the probability that they will move together into the same sex cells becomes larger. And this is why linked genes is an example of non-Mendelian genetics. It doesn't accept the law of independent assortment. It is non-Mendelian. Okay, so hopefully that made sense. If not, we're still going to be talking about this a little bit, so I am open to more